Hi student, I am Dr. D.S. Thavre. Please wait 10 seconds. I share the screen of today's lecture in front of you. Welcome to the third lecture of course of fifth semester, that is path 3.4. Title of this course: Diseases of the field and horticultural crops and their management. Today we discuss regarding pythium causing diseases in horticultural crops and vegetable crops. So, first of all, we know the general characteristics of Pythium genera. Pythium is a soil-borne pathogen. It is a genus of class Oomycetes. Pythium is a parasitic in nature and reproduced by sexually as well as asexually method. So, Pythium is a most destructive nature of pathogen causing damping of and stem rot diseases is produced in different horticultural and vegetable crops. So, this is the three most important diseases seen in papaya, tomato, and ginger caused by Pythium genera, that is, soft rot of papaya caused by Pythium apinidermatum, damping of tomato caused by Pythium divarianum, and rhizome rot of ginger caused by Pythium apinidermatum. Now we discuss one by one. So first of all, we discuss regarding soft rot or stem rot of papaya. So we go to the first point, that is a symptom. Pythium is a soil-borne pathogen and survival in soil. Suppose the farmer can sow the seed of papaya in nursery bed area. So under favorable conditions, the Pythium pathogens germinate under the favorable conditions and infect to the papaya seeds, means the mycelia of Pythium grown on the outer surface of the papaya seeds. So these infected seeds cannot germinate and produce the seedlings, means these seeds is totally rotted and do not have ability to develop the seedlings. But sometimes the infected seeds germinate and to produce the seedlings, means these emerge out the seedlings from the soil line. So number of the symptoms also be developed on these germinating seedlings. So see the basal portion of these engaged seedlings on which brownish lesions are developed at the collar portion or just on this stem just above the soil lines. So these brownish lesions girdle the engaged stem. So these infected seeds is totally rotted under favorable conditions. So in this way, number of the seedlings get collapsed and dries in nursery bed areas. So this symptom is known as the pre-emergence rotting or pre-emergence type of the symptoms. Then second symptoms, that is post-emergence rot, means during the development of the seedlings or on the mature papaya plants, so number of the symptoms are seen. So basically the brownish lesions, means water soaked spots, is seen on the collar portion of these stems. So these water soaked lesions turns to the brown in color. So brownish lesions is develop and girdle around these stem portions. So this fungus develop under the favorable conditions and spread these pathogens in upward directions and also the downward directions, means infection seen on the above parts of this plant and underground parts of these plants. So just to see these infected plants, means the produced leaf are yellow in color, or after some days these leaf are dries, so number of the leaf is defoliated from these plants. So these plants is toppled over and dies because disintegration of parenchymatous tissue of the stems, means rotting is extent above and below the stems. So such infected stem or plant is uprooted from this place and cut the internal tissue of the stem. So honeycomb fashioned appearance type of the symptoms is seen of this section. So this is the blackish parenchymatous tissue are seen, means this tissue is totally rotted. So simultaneously number of the plants to be infected by these Pythium pathogens. So the number of the plants infect and seen in particularly papaya fields. So this is the symptoms. Then goes to the next point, that is a causal organism, that is a Pythium aphendermatum. So Pythium aphendermatum fungus reproduced by sexually as well as asexually. So initially this Pythium developed the mycelia, means synocytic hypa is developed under favorable conditions and to produce either is a sexual spore fruits or asexual spore fruits, depends upon the temperature developed in particularly atmospheres. So this is the disease cycle or life cycle of Pythium. So this is the somatic hyphae. Somatic hyphae germinate under the favorable conditions. So finally produces the oogonium, means male gametes, and female gametes, and antheridium means male gametes. So these oogonium and antheridium fuse to each other under plasmogamy and meiosis. So finally develop the oospores, means two-walled spherical oospores is developed by sexually manner. So these spores germinate under the favorable conditions, means when the temperature is maintained in atmosphere to exceed the 28 degrees Celsius. So finally these oospores also develop the somatic hyphae. Means in this way, complete the life cycle of the pythium by sexually manner. Means finally develop the oospore. But sometime means temperature is maintained below the 28 or below the 20 degrees Celsius. So the somatic hyphae germinate and to produce the spore fruits, that is the sporangium. So the sporangium develop the sporangia, means after maturation the wall of the sporangium release the sporangia. So these sporangia again germinate and to produce the zoospores. So zoospores is a biflagellate type of the spores and always having the kidney shape. So the zoospores is produced from the infected lesions and halon on the another healthy one plants. So in this way, the spread of the disease from infected plant to the healthy plants through asexual spores, that is a zoospores. Means in regarding the perpetuation, primary infection is takes place through the oospores always present in the soil. This is the one possibility. Then another possibility means suppose the infected seeds sown in the infested soil having the pythium, so 100% this is the produced. So this is the two ways for initiate the inoculum and infect to the healthy one plants. And spread of the disease means secondary source of infection, that is a sporangia and zoospores mainly spread through water or mainly through also airbornes. Okay, then next point, favorable conditions. So this pythium mainly infect to the younger plant as compared to the mature plants, and these symptoms is majorly seen in June month to the August month. Then another favorable conditions, that is excessive moisture, then uncontrolled irrigations, means apply the more than 5 irrigation to particularly papaya in a particularly 1 month period. So 100% this is produced. Then heavy rainfall and most suitable condition, that is waterlogged soils. So these parameters is favorable for disease development. So this is the some information about the foot rot or stem rot of papaya. Then how to control this disease by using the chemical method or by using the cultural method or any methods. So first management points, that is a badly affected plant should be uprooted and burned. Then seed treatment with Captan or Thyron or Ballistin at the rate of 2 to 4 gram per kg of seed. So finally, reduce the disease. Then third one, seedlings should be raised on well-drained nursery area because this disease mostly seen in waterlogged soils. Then food management, that is the diseased seedlings should not be used for replanting. Suppose already disease is produced in main field. So drenching the base of this infected stem of the plant with borax mixture at the rate of 1% or Captan 0.1% or copper oxychloride 0.25% or recently most effective fungicide, that is metalaxyl, at the rate of 0.1%. So this is the different fungicides is effective to reduce the incidence of disease. Instead of the chemical methods, some biological method is also effective. Means the seed treatment with trichoderma viridis powder at the rate of 4 gram per kg of seed, means to protect the seeds from the fungal infections by applying the biological agent as well as the chemical agent or fungicides. So this is the basic informations about stem rot or foot rot of the papaya. Okay, then we goes to the second disease, that is the damping of tomato. So this damping of disease majorly seen in all the vegetable crops grown on the nursery bed areas as well as in the main fields. So this pythium infect to the tomato, so produces the two types of the symptoms. That is, one is the pre-emergence rot and second is the post-emergence rot. So see these diagrams. So this is the healthy one plants and this is the infected plants. Means the spores is attached to these germinating seeds as well as these germinating seedlings. So these germinating seedlings is killed before they reach to the surface of the soil. Means radicle and plumule is completely rotting, or sometimes the brownish spots or lesions is seen on these collar portions. So these pathogens spread in upward direction or maybe downward directions. So finally these seedlings are totally rotted. So this symptoms is known as the pre-emergence rot. Then see these second pictures in which the symptoms is developed on the well-developed tomato plants. Means initially the brownish spots is seen on the basal side of this stem of the tomato plants. So these brownish lesions is spread to the upward direction as well as the downward directions. So these pathogens develop the mycelia and symptoms, means girdle these brownish lesions around these developing stems. So finally the weakened tissues is seen in this point of infection. So top of the plants is collapsed from this point of infections. Means in this way the total plants is collapsed and rotted. So this symptom is known as the post-emergence rot. Okay, then causal organisms, Pythium diparinum. Pythium diparinum fungus develop the mycelia. So this mycelia is always haline. Haline means colorless. Cynocytic. Cynocytic means non-septate. Branched type of the mycelia is developed. So this mycelia develop under the favorable conditions and to produce the asexual spores, that is zoospores and sporangia. by forming the sporangiophore. So just to see this life cycle. So this is the sporangiophore and sporangia. So number of the sporangia is developed inside the sporangium. After maturation of the wall of the sporangium, release the sporangia. So again the sporangia germinate under the favorable conditions and to produce the zoospores. So the zoospores germinate under the favorable conditions and infect with the healthy one plants. So in this way, the spread of the disease from infected plant to the healthy plants via the asexual spores, that is sporangia and zoospores, is mainly spread through water. Then sexual spores means the after fusion of male and female gametes, that is oogonium and antheridium, so finally develop the oospores, and these oospores goes to the dormant conditions for a several years in soil. After favorable conditions, these oospores germinate and to produce the germ tubes. So finally, develop the somatic hyphae. Means in this way, primary infection takes place to the newborn healthy one plants in tomato. Then last point, that is a control measures. Avoid waterlogging and low-lying land because this pythium is highly developed in waterlogged soils. So avoid this waterlogged soil for cultivation purpose. Then use seed from disease-free area because the seed is a primary source of infections. So avoid these infected seeds for sowing purpose. Then seed treatment means to protect the seeds from the fungal infection by applying Thiron or Captan or Bavistin or organomercurial compounds at the rate of 2 to 4 gram per kg of seed before the sowing. Then after treating the seed, you can sow in the main fields as well as in the nursery areas. Then soil treatments, because this pythium is survival for a long life in soil. So eradicate the spores present in soil by applying some soil treating fungicides. For example, formaldehyde means you can use the formalin 40% diluted with the 50 times with waters and just applied to the nursery bed areas, or spray 1% Bodax mixture 15 to 20 days before the sowing, or sow the seed on raised bed areas and avoid overcrowding of seedlings because these pathogens is mainly spread or developed in waterlogged soils. So always you can sow the seed of tomato in well developed raised beds. Sometimes this pythium can also be eradicated by using biological treatments. Means you can use the trichoderma powder for seed treatment at the rate of 4 gram to 6 gram per kg of seed, or they firstly prepare the the trichoderma solutions, and this solution is a drench around the root of the infected plants. But this method is possible in only limited area, or generally this practice is suitable for in nursery bed areas, not suitable in the uh, open field. Okay, so this is the two disease. Then last disease, that is the rhizome rot or soft rot of ginger. So this Pythium genera infect the rhizome plants and develop the symptoms on leaf of these rhizome plants, ginger plants, or develop the symptoms in basal side of the stem portions, as well as develop the symptoms on underdevelopment of the roots as well as rhizome portions. So initially, these pathogens develop the symptoms on basal side of the stems, means at the collar portion or stem portions having the brownish lesions are seen. So these brownish lesions just spread in upward direction as well as downward directions. Means suppose these pathogens move in upward directions. So number of the symptoms seen on the infected plants. So just to see this individual leaf of these plants. So yellowing symptoms is majorly seen. So this yellow symptoms it starts from the tip of the leaf to the downward directions. Means the yellow symptoms is developed from the margin of this leaf to the downward directions. So in this way, number of the leaf get infected and showing the yellowish symptoms. But after some days, these leaves are totally dry and hang down to the pseudostems. So these symptoms generally seen on the upper parts of the plants, but mainly symptoms is developed on the underground parts of the rhizomes. So the initially water soaked translucent spots is developed on the underground root portions, means on the rhizome portions. So these water soaked translucent spots turns to the brown in color. So brownish lesions it enlarges to the entire surface of the rhizomes. Means these lesions gradually decompose, forming a watery mass of putrefying tissue enclosed by the tough rind of the rhizomes and also the vascular strand of the mycelium. So, the roots arising from the infected lesion or region of the rhizomes also show the typical softening and rotting of the tissues. Means these two types of the rotted symptoms is seen in ginger. That is the first one dry rot and second one wet rot. Dry rot means the infected rhizomes having the tissue become dry in nature, but in the wet rot, the infected tissues having some of these moisture is present, means wet rot symptoms. So this rhizomes is totally rotted. So this is the symptoms developed by the fungal pathogens. But in ginger, sometimes bacteria is also infect and develop the symptoms on rhizome portions. So how can identify the bacterial infection and fungal infection on rhizome portion of the ginger? So only one points to be considered, that is a smell. Means suppose odor smell is emitted from these infected rhizomes. So this infection is produced due to the bacteria. But in fungal infection, no any smell is emitted from these infected rhizomes. So in this way, whole plants is rotted. Suppose this infected dried ginger plants is uprooted from these fields. So easily uproot these plants. So this is the indication of these rhizome rot symptoms caused by the PTM. So this is the overall symptoms is developed on the ginger plants. So this is the healthy one plants and this is the infected plants. So infection generally seen on this leaf portion, seen on these younger rhizomes, and this is the mature rhizomes. So typical symptoms seen on this leaf, this stem portion, and this developing the rhizome parts. Okay, then last point, that is a causal organism, Pythium aphendermatum. So Pythium aphendermatum fungus is always haline in nature and produces the branched hyphae and also non-septate in nature. So this fungus germinate under the favorable conditions. Means suppose this is the infected plants and this is the infected rhizomes. Means after harvest, some of the crop residues or rhizomes, infected rhizomes remain present in the soil, means debris in soil after the plowing operations. So number of the hypa and mycelia is survival for a long durations in soil, but under favorable conditions, the somatic hyphae develop the Ubonium and Anthridium. So finally develop the oospores, means two-walled smooth oospores is developed and these oospores fallen on the healthy one leaf of the ginger or stem portion of the ginger or in fact the developing rhizome parts. So these spores penetrates into the healthy one host tissue. So this healthy one host tissue become infected. Is in this way, primary infection takes place through these oospores and this healthy one plant become infected. So these infected plants already develop these spores and these spores, that is sporangiospores and zoospores, means the somatic hyphae develop the spore fruits, that is known as the sporangium. So inside the sporangium, number of this sporangia is developed. So this sporangia liberate through this wall and develop the zoospores. So this zoospores is a biflagellate. Biflagellate means these zoospores is always float on the water through these flagellate portions. So these zoospores directly infect with the healthy one plants. So this healthy one tissue become infected. Means in this way, the disease can spread from infected plant to the healthy plants through zoospores and sporangium. So this is the perpetuation of this Pythium genera. So this is the sinusotic hyphae. Means these non-septate or cross wall is not seen in these developing mycelia. This is the oospores. Oospore is a sexual spores develop after the fusion of male and female gamete, and this is the sporangia. Means sporangia is a sexual spores develop inside the sporangio or sporangium. Then favorable conditions, these ginger crops is a rain-fed as well as irrigated crops. So warm and humid climate is favorable to produce the disease in ginger. Then optimum temperature range, that is a 34 degrees Celsius, this temperature range is favorable for germination of the oospores as well as the sporangiosperms, that is the zoospores. Then optimum relative humidity means the 60 to 80% relative humidity is maintained in atmosphere. So this is the number of the parameters to be considered for development of the pythium in ginger plants and produces the rhizome rot symptoms. Then last point, that is a control measures against the pythium pathogen in ginger crops. So primary infection is always takes place through infected rhizomes. So before the actual planting of rhizomes or selection of rhizomes, you can consider the some points, that is a rhizome is always disease free from the rhizome fields. Then these rhizomes is always dipped in already prepared fungicide solution, that is a carbendazim solution or mancozeb solution for 60 minutes, means 1 hours. This treatment generally done after the harvest of the rhizome in the main fields, because these rhizome rot symptoms is also seen on the rhizomes in storage. So avoid the rotting symptoms in storage conditions. So immediately harvested rhizomes is generally deep in carbendazim solution for 1 hour. Then follow at least 2 to 3 year crop rotation with non-host crop, because this pythium is a soil-borne pathogen. So eradicate the pythium by adopting the crop rotation practices with non-host crop. Then apply neem cake, that is a 250 gram per meter square area, as well as the lime as a soil amendment to reduce the pythium inoculum in soil. Then another management practices generally done by the farmers, that is the soil drenching with antagonistic species of trichoderma, that is the Trichoderma viridi, Trichoderma harjanium, Trichoderma hamatans, generally applied around the infected root of the ginger, that is a 40 gram per meter square area. So in this way, you can kill the spores or inhibit the growth of the pathogens around the roots of the ginger plants. Then drench the infected plant with copper oxychloride 0.3% or borax mixture 1% during rainy season to prevent the further infection of the pathogens. Means in this way you can apply the IDM practices to ginger crops for avoiding the rhizome rot disease. So today's already we discussed the three diseases in papaya, that is soft rot, damping of tomato, and rhizome rot of ginger. So these three diseases is mainly caused due to the pythium pathogens. So this is the end of the today's lecture. Any doubt, please ask to me personally or call to me personally. Thank you.